The overall goal of the following experiment is to introduce oncogenes or other potentially hazardous genes into human cells using ecotropic lentivirus. This is achieved by producing pantropic lentivirus encoding the mouse retrovirus receptor, as well as ecotropic lentivirus encoding the gene of interest. Cells are transduced with the mouse retrovirus receptor, which makes them susceptible to transduction with ecotropic virus. Next, the cells are transduced with ecotropic virus in order to introduce the oncogene, using polymer-complexation with polybrine and chondroitin sulfate to enhance transduction. Results are obtained that show selective transduction of human cells with ecotropic lentivirus based on fluorescent microscopy and FACS.
The main advantage of this technique over existing methods, such as gamma-retroviral transduction, is that lentiviral particles can more effectively transduce many cell types, including non-dividing cells such as stem cells.
This method can help answer key questions in the field of cancer and stem cell research, such as understanding the function of potential oncogenes.
Before beginning this procedure, consult your institutional safety official and follow their recommended safety guidelines. Begin with a culture of healthy, rapidly growing 293 T cells. Working in a tissue culture hood under aseptic conditions, resuspend 5 × 10⁶ 293 T cells in 10 ml of antibiotic-free 293 T medium. Pipette the cell suspension into a 10 cm tissue culture plate and incubate the plate overnight in a tissue culture incubator at 37 degrees Celsius and 5% carbon dioxide. The next day, begin transfection in the late afternoon. First, collect an aliquot of Optimem and the transfer, packaging and envelope plasmids and allow these to warm to room temperature. Pipette 375 microliters of Optimem into a microcentrifuge tube and then add 25 microliters of Fugene HD transfection reagent. Ensure that the Fugene reagent is pipetted directly into the media and does not touch the side of the tube. Next, in a second microcentrifuge tube, combine 5 micrograms of the transfer plasmid, 3.75 micrograms of the packaging plasmid, and 1.25 micrograms of the envelope plasmid in the required amount of Optimem to bring the volume to 100 microliters. Then, add the Optimem Fugene mix to the tube containing the plasmid mixture and incubate at room temperature for 20 to 30 minutes. During the last few minutes of the incubation, very gently aspirate the media from the plate containing the 293 T cells and replace it with 10 ml of fresh, antibiotic-free 293 T medium. Finally, add the Fugene plasmid mix dropwise into the media of the 10 cm plate. Place the plate in a tissue culture incubator set to 37 degrees Celsius and 5% carbon dioxide and incubate overnight. The next day, very gently aspirate the media from the plate and replace with fresh, antibiotic-free 293 T medium. Then, replace the plate in the tissue culture incubator and incubate for a further 2 days. 3 days after the transfection, aspirate the media containing the virus from the plate and filter it using a 0.45 micron low-protein binding filter. After filtering, titer the virus. To titer virus, make serial dilutions of a fluorescent control virus in fresh antibiotic-free media with 6 micrograms per milliliter polybrine and use these dilutions to transduce cells of the appropriate cell line in a 24-well plate. The next day, change the media to fresh antibiotic-free media and then continue the incubation for a further 2 days to allow the cells to express the fluorescent protein. Use FACS to determine the fraction of transduced cells for each viral dilution. Then, calculate the titer in transforming units per milliliter based on dilutions that yield less than 15% transduction to minimize multiple transduction events. The titers of other non-fluorescent lentivirus produced in parallel are presumed to be similar to that of the fluorescent virus. To transduce target cells, begin by selecting a multiplicity of infection greater than 2 to ensure that most cells will be transduced. First, dilute the viral supernatant to the required multiplicity of infection in fresh, complete culture medium, supplemented with 6 micrograms per milliliter of polybrine to a final volume of 1 milliliter. Aspirate the media from a 35 mm dish of target cells and add 1 milliliter of diluted viral supernatant. Incubate in the tissue culture incubator for 24 to 48 hours to allow sufficient expression of the SLC7A1 receptor. If desired, use antibiotic selection with blastocidin to select for stably transduced cells expressing SLC7A1 according to the instructions in the written procedure. To begin toxicity determination, prepare a stock solution of 20 mg per milliliter chondroitin sulfate and a stock solution of 20 mg per milliliter polybrine in water and sterile filter. Excess stock solutions may be stored at -20 degrees Celsius until needed. Next, determine the number of 96 well cell cultures needed to accurately determine polymer-complexed toxicity using the MTT assay. At least 6 concentrations of chondroitin sulfate and polybrine should be tested in triplicate. In addition, untransduced cells and cells grown in serum-free medium should be included to provide a baseline for healthy cells and to control for growth arrest, respectively. Once the number of required wells has been ascertained, plate 5,000 target cells in each well of a 96-well plate. In parallel, plate cells in a 24-well plate for microscopic or FACS-based analysis of transduction efficiency in each experimental condition. Allow both sets of cells to grow overnight. The next day, thaw an aliquot of the chondroitin sulfate stock solution and an aliquot of the polybrine stock solution. Then, dilute virus-encoding GFP in antibiotic-free media containing 6 micrograms per milliliter polybrine to a multiplicity of infections sufficient to transduce around 10 to 50% of target cells per well. Ensure that there is enough diluted virus for each test condition plus control wells on each plate. Label a set of tubes with each chondroitin sulfate and polybrine polymer concentration to be tested, then aliquot enough of the viral suspension for each condition in triplicate into the tubes. Next, pipette the required volume of chondroitin sulfate and polybrine stock solution to make up the test concentrations into each of the tubes in succession. Immediately after the addition of chondroitin sulfate and polybrine, flick the tube to mix. Allow the virus chondroitin sulfate polybrine mixtures to incubate at room temperature for 5 minutes. The mixture may become cloudy as viral precipitates form. Following the incubation, aspirate the media from the wells of the 96-well plate and then add the virus chondroitin sulfate polybrine mixture to the wells as planned. Also pipette control and serum-free media into the appropriate wells. Repeat the process for the 24-well plate. Incubate overnight in the tissue culture incubator. Next, aspirate the virus-containing media from the plates and replace with fresh medium. Return the plates to the incubator for a further 48 hours. After 48 hours, remove the 24-well plate and use FACS to analyze the transduction efficiency of each chondroitin sulfate and polybrine concentration. The following day, aspirate the media from all wells of the 96-well plate and replace with 100 microliters of MTT solution. Culture for 3 hours in a humidified incubator to allow MTT to be reduced in the mitochondria of metabolically active cells. After 3 hours, aspirate the MTT solution and replace with 200 microliters of solvent solution. Incubate the plate for 2 hours at room temperature or until all of the purple MTT formers and precipitate is dissolved. Read the absorbance on a microplate reader at 570 nanometers, subtracting the background reading at 690 nanometers. The optimal polymer concentration will produce maximum enhancement of transduction as determined by FACS with minimal effect on metabolic activity as determined by MTT. For HDFs, 100 micrograms per milliliter of chondroitin sulfate polybrine is the optimal polymer concentration as determined by this method. First, prepare enough 100 microgram per milliliter chondroitin sulfate polybrine viral supernatant mixture to transduce the required number of 35 mm dishes of SLC7A1 receptor-expressing HDF cells. Incubate the mixture for 5 minutes to allow polymer complexes to form. Then, aspirate the media from the receptor-expressing target cells and replace with the viral mix. Incubate overnight at 37 degrees Celsius and 5% carbon dioxide. After removing the viral mix, wash the surface of the cells twice with PBS to aid in removing viral complexes. Complete removal is not necessary, as no adverse effects on cell health have been observed from residual polymer complexes. This fluorescence microscope image illustrates efficient transduction of human sarcoma cells stably expressing SLC7A1 with ecotropic virus when using fluorescent control vectors to monitor transduction efficiency. Quantification of fluorescence using FACS analysis demonstrates a transduction efficiency of greater than 90%. Transduction rates of HDFs are generally lower because the cells have not been blastocidin selected for receptor expression. Titers of ecotropic lentivirus are generally 10 to 20% of VSV pseudotyped virus when measured on SLC-HSC. The MTT viability assay allows sensitive detection of growth arrest in target cells. As seen here, transduction with virus plus concentrations of chondroitin sulfate polybrine up to 800 micrograms per milliliter have no effect on HDF metabolism. FACS analysis of HDFs transduced with virus plus various concentrations of chondroitin sulfate polybrine shows enhancement of transduction compared to polybrine alone. Maximum enhancement occurs at 100 micrograms per milliliter chondroitin sulfate polybrine, several-fold lower than previously reported values. Thus, it is important to optimize conditions for any given cell type. Complexation with chondroitin sulfate polybrine enhances the observed titer roughly threefold in SLC-HSC. In practice, this yields a greater effect on transduction efficiency at low virus concentrations than at higher concentrations, which is most likely due to multiply transduced cells and receptor saturation at higher virus concentrations. Transduction with ecotropic virus is specific for cells expressing murine retrovirus receptor. When transducing unmodified human cells with ecotropic virus, fluorescence greater than the untransduced background has not been observed as seen by FACS analysis. Microscopically, HDFs show no transduction in the absence of receptor, while pre-transduction with receptor results in fluorescent cells.
When attempting this procedure, it is important to remember to verify that the virus will not transduce unmodified human cells.
After watching this video, you should have a good understanding of how to use ecotropic lentivirus to deliver potentially hazardous genes to human cells, including the use of polymer complexation to enhance transduction.